Hello. Ilagam Friends Group. People. Hello. Huh? Hello.
Sir, 1 minute, sir.
Okay, madam. Okay, madam.
Sir, let me start the program. So we are waiting for the students. Sir?
Hello.
Sir, let me start the program, sir.
Yeah.
Okay. Aishwarya?
Yes, ma'am.
Yeah, start the program.
Good evening to everyone who all are gathered here. I'm glad to welcome you all for innovation and development in research techniques and IPR research to impact on innovation and knowledge transfer conducted by SHEE Club and IIC of Prince Sri Venkateswara Arts and Science College. Let's begin the program with Tamil Thai Valthu by Krishnaveni. Krishnaveni? Krishnaveni? Okay, Aishwarya, continue. Yes, ma'am. Now I would like to call upon Sandhya to deliver the welcome address.
Sandhya?
என்னோட ஸ்கிரீன் இங்க இருக்கு.
Sandhya? Ma, can you? Sandhya?
Yes, ma'am.
Yeah, start.
Good evening, everyone. It's my pleasure to start this auspicious occasion by heartily welcoming everybody. It's my great pleasure in welcoming you all in this webinar session. The topic is Innovation Development in Research Technique and IPR Research to Patent Impact on Innovation and Knowledge Transfer conducted by SHEE Club and IIC of Prince Sri Venkateswara Arts and Science College. I'm profoundly delighted to take an opportunity to welcome our chief guest for this evening. First, I would like to welcome our chief guest, such a noble personality, Dr. S. Gopalakrishnan, He Pro Assessment Solution Private Limited, Chennai, and also welcome our another chief guest, eminent speaker, Dr. Dj Mukesh Kumar, Principal Scientist, Avijan Biotech, Chennai. I deem it as a great pleasure to and privilege welcome our prominent chairman, Dr. K. Vasudevan, who always ready to walk miles for the cause of education. I take great honor to welcome our energetic vice chairman, engineer V. Prasanna Venkatesh, who knows himself how to keep pace with modern technology and use them for good. It is a matter of great pleasure and honor for me to welcome our beloved principal, Dr. M. Uma Bhaskar, who gives lots of encouragement to all the teaching staff and students to give the best. I am proud to invite our SHEE Club coordinators and IIC members, dignified faculties, and also our brilliant students and participants. I welcome you all once again. I'm sure that we all get enlightened with this webinar and benefited. Thank you.
Thank you, Sandhya. Now I welcome Dr. K. Sindhu, Ambassador of IIC, to share a few words about IIC.
A very good evening and warm welcome to one and all present here. Ministry of Human Resource Development has established Institutions Innovation Council to foster the culture of innovation among all higher education institutions. IIC is a faculty expert student council which takes on multiple activities to promote innovation and entrepreneurship. The primary mandate of IIC is to encourage, inspire, and nurture young students by supporting them to work with new ideas and transform them into prototypes. The main focus of IIC is to create a vibrant local innovation ecosystem, thereby contributing for the development of entrepreneurs in our country. So the main focus of IIC is to identify and reward of student innovators. So it focuses to organize workshops, seminar, and interactions with entrepreneur investors and professionals. So the theme of our college IIC is Imagine, Innovative, and Invention. So everyone gets the opportunity to become an innovator through various motivation programs of IIC. Thank you.
Thank you, ma'am, for your valuable words. I would like to call upon Dr. K. Deepalakshmi, coordinator of SHEE Club, to deliver a few words about SHEE Club.
Yeah, good evening to one and all present here. It's my pleasure to take this opportunity to recite about SHEE Club. The club was inaugurated for the welfare of students and public to give awareness and enlighten their minds towards the environment founders. In this way, in our SHEE Club, we created so many awareness programs about safety, health, environment, and empowerment in day-to-day life. Some of our awareness programs which progress and sparkle the mind of the students and public. They are eradication of plastics, planting trees in the project of Go Green, conservation of biodiversity, health awareness program, culture and traditional programs, empowering the students in organic farming and subject-related empowerment programs. There are so many projects we done. These are some of the snaps we have taken during that program and awareness program. And this will be helpful to each and everyone to encourage themselves in eco-friendly activities to protect our environment. So as one of the motto of SHEE Club is empowerment the students. So in this way, we are conducting these IIC programs to encourage the students towards the entrepreneurship. Thank you.
Thank you, ma'am. Now I would like to call upon G. Rita to introduce about chief guest.
Good evening, one and all present here. I am highly honored and feel privileged to have been given the opportunity to introduce our guest in this webinar section. Our exalting person, Dr. S. Gopalakrishnan. He completed his postgraduate in preliminary studies on the food and feeding habits of some freshwater fishes at Kadaperi Lake at Pachayappa's College. Master of Philosophy in effect of zinc on fertilization and early development of sedentary polychaete hydroids, elongates at the Presidency College. Doctorate in effect of copper and aluminum on reproductive behavior, gametes, fertilization, and early development of sedentary polychaete hydroids, elongates at the Presidency College. He worked as a research associate, postdoctoral fellow, visiting scientist, visiting scholar, postdoctoral research associate. He is running at ePro Assessment Solution Private Limited, Chennai. He has published many papers. Thank you.
Thank you, Rita. Can you all kindly turn on your camera for taking clicks? Kindly all of you turn on your cameras.
Okay, Aishwarya, continue the program.
Yes, ma'am. Thank you. Please turn off your videos. Now I welcome our chief guest, Dr. S. Gopalakrishnan, to take up the session.
Sir?
Yes, good evening, everyone. Am I audible?
Yes, sir.
Okay, good. Let me share my screen.
Yes.
Okay. Audio கேக்குதா? Yeah. Am I audible?
Yes, sir.
Okay. Myself, Gopalakrishnan, and I'm very thankful for giving an opportunity to give a talk in Prince Venkateshwara College. And they have given enough introduction about me. So let me go directly to the sessions. And today I just going to give a talk on innovation and development in molecular techniques. I'm not going to bore you by telling more things about the innovation development, because and nowadays these young guys are more advanced in knowing these things than us. So let me, what is innovation? So all we know about innovations, and some few of them is confused with about the innovation as well as the invention, because there is a two term, invention as well as innovation. It is different term, but most of us will confuse with that one. So what is innovation? Is that artistic creativity, or invention, or bright idea, a scientific discovery or technical solution, and simple incremental improvement? I don't think all these things comes under innovation, because innovation is something different, uh, altogether what they have shown in the slides. That is, your idea, that idea also need to be a novelty, and that novelty brings your idea, and that idea need to give a invention. That inventions need to be you gear leads to one innovative items that to be used for the public use, and that maybe leads to some other process, and again it will be a cycle change. And so as a term, we can see innovation is a process followed to obtain something new that solves a specific problem and can be utilized. So I told already, what about invention and innovations? Invention is something different. Like you invent, for example, in those days, wheel has been invented, like to move anything on the wheel. So based by utilizing the wheel, they like innovated a car, like a four-wheeler. Similarly, in the world nowadays, we use internet. So internet is like innovation, like invention. But by the use of internet, Facebook, WhatsApps, and so many apps are like by using the internet, they innovated and they connected the people like anything. So this is the difference between the internet invention as well as the innovations. And what about research and innovations? Like what do you mean by research and what cause innovation and research? Like innovation and research are like bonded together. Like research and development and innovations are reality very close to each other. All they present different concepts, even though it is a different concept. But like because research is something different, development is different, innovation is different, but together they will join to proceed for one. And this is like a cyclic one. Like you share the knowledge to get it innovated, and by innovating something, you'll be earning and put them with the same money for the research. Again, you share the knowledge to the public. And research types, there are two types, like basic research as well as the applied research. Like maybe I just give something about the basic research, because most of the students are attending here. What is research? What is good research? What's different types of research? The key process in conducting research, and the general principle and common pitfalls in experimental designs. What are the, like once if you completed the research, you will be awarded, like when you're doing the PhD, you will be awarded the degree, philosophy of doctorate. Then what is the philosophy in research? What is doing that? Like in like Oxford Dictionary, the study of truths and principle of universe, life and morals, and of human understanding of these. These is the terms for the philosophy in Oxford Dictionary. But whether it is true in research, then what is research? Research is the like collection of data, analysis of data, interpretation of data. Is this risk? Because when you are doing the polling, you'll be collecting more information and how many people they poll to select a president or like MLA or MP or anyone, or even the school leader or college leader. Is this conduct of polling is the survey or research? Yes, of course, it is a kind of research, because you are searching someone to lead from the group. So it is a kind of research. So what is then good research? Good research, it should be like novelty and originality, like it has never done before, advance our knowledge and understanding on the aspect you proceeds. Is this enough for the good research or anything else? Good research in terms of generalization. For example, Watson and Crick developed the DNA structure. It's a kind of research on advancement in DNA technology. But based on that, so many things are advanced in recent years, as even though like more than 5 to 6 decades ago, the DNA structure was found. Till now, it has been established, like the genetic code for all the living organisms based on the DNA structure we delivered, and it explained the replication and the protein synthesis. So the research which 60 years back done by Wasson and Crick, even till now it has been utilized. So this is a generalized research and it comes under goods research. And good research can also be their prediction. For example, the Nobel laureate like Linus Pauling found, like he has given a Nobel Prize for the chemical bonding. Nowadays you will be using HCl. The student may be knowing is as a HCls. Whether you think about how it forms, like because the H plus and the Cl minus combine with an polar covalent bond to form the HCls. This prediction is given by Pauling, and he had been given the Nobel laureate for this one. And this is a good research. Until now, we are using that one. And good research also need to be a wide applications. For example, in like 30 years back, Carey Mullis found out the PCR technique, and using the PCR technique, we may know like millions copies of DNA will be produced in an hour. And this wide application, even right now, it is being used, and even in recent days, like for identifying the disease of the corona, we are using the same technique in advanced manner, like the real-time PCR. If this real-time PCR is based on the polymerase chain reaction only, and this is the it has invention for the biotechnology in those days, the PCR technique, but it has wide applications till now. And the good research has to, because when you are publishing something, it has to reach several peoples. And PCR technique, when you publish, it has reached till now like nearly 28,000, and the HI index it got us 16. And recent years, even like last year, the Nobel Prize has been delivered for the David and Anthem for the subject in the physiology, and these all have the wide applications. All these comes under the good research. So good research means it has to have wide applications to everyone. For example, Microsoft Windows and a search like engines like Google. You see, like Microsoft Windows, without Microsoft Windows, we most of the 99% of the computer persons use the Microsoft Windows. So when it is invented, it may not be known whether it has reached this much level. So it has the wide application, similar as the Google invention. So vital elements of good research is the originality, novelty, able to make generalization, able to make prediction, and wide application. Ask yourself this question: how about my own research project? Because you'll be now, some of the students may join a PhD program, or some of them may, in coming years, they may join, but you have to ask about yourself whether you're going for the, what kind of project you are going to join. So what the type of research then? Fishing expedition, discovery, technique development, hypothesis testing. All these are different types of research. Like fishing expeditions, if you go to the Chennai Harbor and stand in front of the Chennai Harbor and check how many species of fishes are there in Chennai Harbor. If you ask the questions, you'll have so many answers because everyone knows there are thousands kind of species out there, but only few are known very well. But in that, you need to know what's difference you make in that one. So just ask a question like, how many species of fishes are there in Chennai Harbor? Then fishing expedition, there will be like, what is the level of any pollutant in the coast, Chennai coast? The first question you ask, what kind of fishes are, and the second question is you are going to ask what is the pollutant and what is the level in the Chennai coast. So what you're going to discover on this one, like which species is most sensitive to the pollutant. Earlier you asked how many species are there. Then the second question you ask, what are the pollutant present and which species is more pronounced for this kind of pollution and which is like most sensitive for this pollutant. So the another factor, like what is the optimal temperature for the fish and the pollutant and the optimal pH or salinity for this pollutant to exist there. So these things comes into your mind for the types of research. Then what are the facts you're going to find based on this? For example, you need to discover, like, if you like, what are the numbers of the proportion of yellow and white or on my dog? You'll be having dogs in your like homes. You just very curiously you just see how many yellow has and how many white has in your dog, and this is a kind of research, right? And that you just develop a technique to find out these things, the advanced technique to find out these things, like then find a like a hypothesis. Can a drug A increase the blood pressure on the fish, and a chemical can increase the reaction rate? That is, I'm talking about here, because when once you went for the fishing, like how many species of fish present in the harbor, then you just ask the question about the pollutant in the fish. Then you ask what kind of pollutants will harm the fish. And now you ask the question whether any drug can increase the blood pressure or any biochemical components in the fish or any chemical which increase the reaction rate in that one. So what make you think that the drug, that is the pollutant, can increase the blood pressure? So this is the difference. Like when you're going for the fishing, you may ask, like I'll be going for the fishing because there will be presence of more fish in the sea, rather than, like, this is the generalized term. But if as a scientific, what you have to tell, like since I'll be going for the fish because at the time I like 4 p.m. Yeah, I'm going for the fish. It should be more fish available because the tidal current and the season may be more for the fish to be appear more in that one. So this is the scientific reason you're going. This the difference. Like if you're going for fishing for without any answer, that is generalized, and if you are going with a specific answer, that makes the research difference. So next, so like the discovery can also have a very significant impact, whatever you discovered. Like the penicillin, till now it has been like have a tremendous effect for the illness, and as well as the superconductor, what we discovered earlier in the physics. And the type I already talk about, like how many numbers of as and white or yellow has in your dog. For example, these are the facts finding. Like once if you started doing the research, you need to form a hypothesis and you need to find a fact. For example, if your hypothesis is good, if you are asked the question, like there are good indication that white and yellow hair on your dog and your neighbor dogs all appear a certain proportion. You just check because I told you to find hair, the yellow hair and white hair in your dog. You just go and see is the same as in your neighbor dog. Then you come to one conclusions. So this is a good research, and it's if you want to be a better one, it has to be generalized. For example, you just make this as a fact, like how many white hairs and yellow hairs in the dog which are in my state as well as in India as well as in China are to be more in the world. So it would be excellent if you further ask the questions why this 3:1 ratio found in all dogs. Like you remember how Mendel did this experiment on pea plant and come up with the solutions like 9:3:3:1 in the genetics ends in the right. So when you form a question, it has to be a generalized and it should not be with a certain circle. Then how about now you ask your own question. For example, how about your own research project? Whether you fulfill the fishing expedition, whether you have found any facts in your findings, and technique development, hypothesis testing. So the key process in research is ask a question, building a conceptual hypothesis, and formulate a testable hypothesis, design and carry out the experiment to test the hypothesis, analyze, interpret, and compare the data, and the exploration, generalization, and predictions. And then you need to go for the application of your hypothesis. So some of the innovation in my research career is that like in the field of biotechnology, we invented a microchip like made up of PDMS. This innovation of microchip is to identify the movement of cancer cells because you know the cancer cells spread like anything in the humans as well as in any all the organisms. Like we want to know how these cancer cells spread in different topography. So we found like we want to use a microchip, like we want to develop a tumor on a chip, and this chip has been inserted in the mouse, and we will track how these cancer cells moves from one place to another with light focal image. And this is the like different topography. We use the nano like substrate. This one. Uh, and this one we found that the cells moving in a, if we put a path like a single railway track, it's moving in a same directions, all the cells, it's not spreading. You can see the colored one, like the actin vincillin merged together, like you can see all the cells going in a path. So we found that unidirectional cell switching gate by using the gratings. And this paper has been published in the plus one. So this is the innovation of the microchip in cancer cells. And we want to use a cell, like we want to stop the cell migration. we just switch over to from the narrow one. We make use of like the button type model, whether this button type or semicircle model is stopping the cells by moving, that is whether it can be controlled, the migration of the cells. We inserted and we found the cells moving along the path as the chip going. And this paper has been published in the Society for the Biomaterials. This is the another chip innovations. This is the this one we found we use the another technique, like we compared all the normal chip and the straight one and the semicircle, everyone together to check whether the patterned topography reveal the different types. That is, you have many type of cancer cells in your body. For example, liver cancer cells and lung cancer cells and bone cancer cells, cervical cancer cells, every and each and every organ will have a specific type of cancer cells. So you need to know whether this kind of cells will spread from one region to another, even though all of your organ are made up of same tissue, but it's kind of different formation as there. So we want to know whether that pattern makes this different. So we use different topography for analyze the metastasis of the cancer cells. And this is the another innovations we did. And we also use some innovative technology using ISH, in situ hybridization, and immunohistochemistry technique in the toxicology. We use a single model fish to found a toxicology impact. This is a a single photograph we took, and you can see in this one, we check we use the micro-CT analysis to check whether the mode is due to estrogen. So this we use the micro-CT analysis technique to found this one as well. And this paper was also published in the plus one. And maybe I just want to tell about a person, because when we are doing research, you need to be so much dedicated. And this guy, John P. Giese, he is like a mentor of mine, myself. Like I am traveling along with him nearly 15 years from 2007. And he is one of my collaborator as well. And like why I want to emphasize him here, he's a very very big guy. Now he's a very big toxicologist in the published more than 1,000 research articles, and at the age of 74 is so much active. You see is like citation like nearly 85, and he contributed more than 50 years in the field of toxicology. And it's he is the first person. What is the innovative thing in this is in his research? He is the first person to identify the pollutant in the region of Antarctica. You may be knowing the Antarctica region, no animal can survive till you can see the pollution exists in the surviving animal. So he is the first person to find out the pollutant in the Antarctica. Antarctica region. So he had given a like honored for this in the US, and I it's great honor for me to publish a few papers with Professor John Giese, and I'll be very proud for this. And what I try to tell to the student is like you need to be very dedicated, like John Giese. Whenever I used to talk to John Giese, he used to tell only two mantra, like you have to die for research, because if you are doing some good research, you have to die for research. And another mantra you used to tell, like spit it out rather than keeping in your mouth, because when you found something, don't keep and hide yourself. You just publish it so that it will reach for the public and it will be more application oriented. And now come back to your field. Like in the field of biotechnology, there are so many things need to be achieved. Like biofloc technical technology is much needed right now, because it is right now there is a lack of, even today morning we had a very good, like a very big meeting in the like NIOT on the cage culture activities by government of India. Like there we talk about the cage culture, because there is a lack of tons of fishes going to be happening India because of overfishing. So there is a cage culture program is coming away, and it is not so successful, because if you're using the feed itself getting harmed because of the feed conversion ratio and the toxicology part of the fish, like a feed, and it's reduced the weight. So what is the need of the innovation here? You need to find a technique by using a biofloc, like use of aggregate of bacteria, algae, or protozoa together form a aggregate matter to improve the water quality, which can or which could destroy the food particles and get away the toxicant and give the good water. And this is $1 million project going to happen in few days. It's going to happen definitely. And another thing is microplastic degradation. When you talk about the plastic pollution in throughout the world, microplastic degradation is very, very important. Otherwise, anything cannot survive, and which may all the organisms may suffer like oxidative stress as well as genetic defect as well. So the plastic exists in the environment need to be degraded using the microorganisms. Like you can claim that so many microorganisms are there to degrade the microplastic, but only at the laboratory conditions. When you take to the field, there will be a profound effect all to come together to stop that microorganisms to degrade the microplastic. So you need to find a solution for this to overcome the environmental challenge, because whatever invention or innovation you're doing, it should not only prevent it in the laboratory itself. It should come to the people, and it should be a public, and it should be the application oriented. And the next thing is how we are going to handle the hypoxia factor and the ocean acidification, because due to ocean acidification and the pollution, hypoxia everywhere. If it is hypoxia, the aquatic organisms cannot be survived, as well as the ocean acidification. If the pH is like once, if the ocean, like the global warming happens, temperature will induce ocean acidification is going to happen definitely, and you need to overcome by the way of biotechnology only. So all these are $1 million projects. What about the vaccines for the corona, like 1, 2, 3? Like even we may not know how many types of corona is going to happen or coming in coming days. So we needed techniques to overcome all these problems, and only the biotechnology can give a solution for this one. So now we need to start. Like if not now, then when? If not me, then who? So you need to ask a question before you start your career. So I hope you may not be bored about my talk, and you just start doing some innovation as well as the invention. And this is my, I just want to give my solution to the students. Like your destination is not be any place. It should rather leads to new way of seeing in research. Thank you.
Thank you, sir, for your valuable thoughts. Anyone have any doubts? Kindly ask.
Students have any questions?
I think some research scholars as well as will be there, and maybe the MSc students.
Yes, sir.
Okay, good. But what my kind of suggestion is, the students in recent days, because in the earlier days we used to uh, suffer to get many, if you want to go for see anything research paper, we'll be waiting, we will write to the authors to send the articles. But in recent days you have everything in your hand. So before start any work, you just read. This is my kind advice. And before you start any project, and it's and one more thing is it's very much I'm very much happy to give a presentation, because my daughter is the student of this college. I'm very proud to give a presentation for this college as well, and I thank Dr. Deepa for this, and also the chairman for giving the opportunity. Thank you.
Yeah, thank you, sir. It's a great pleasure. Okay,
Thank
Aishwarya,
you, sir.
continue.
Yes, ma'am. Thank you, sir. Now I would like to call upon D. Muttarasi to deliver the vote of thanks.
Good evening to all who all are gathered here on this wonderful evening. It gives me an immense pleasure to deliver the vote of thanks for this moment to all dignitaries assembled here. First and foremost, I thank our respected chief guest, Dr. S. Gopalakrishnan, Hebrew Assessment Solution Private Limited, Chennai, who despite his busy schedule has found time to grace the occasion. We are grateful to thank our Honorable Chairman, Dr. K. Vasudevan, for his words of encouragement. His able guidance has always encouraged us. I thank our well-wisher, Vice Chairman, Engineer V. Prasanna Venkatesh, for his support, vision, and commitment. And also my sincere thanks to our motivating principal, Dr. Uma Bhaskar, to support for us. I thank our SHEE Club and IIC coordinator for arranging such a motivational and information session. I am so grateful. Thank to our staff members. Last but not least, thank to our students for making a session a grand full success. Thank you.
Thank you, Muttarasi.
My students, wait for the next question. Within few minutes, we will start the next session.
Yes, ma'am.
Um,
Good evening, sir. Aishwarya, let me start
Good
the
evening,
program.
ma'am. Good evening, ma'am. Good evening.
Yes, ma'am.
1 second.
Now I'm going to introduce our another chief guest. I'm profoundly delighted to take this opportunity to introduce our another chief guest for this webinar, our noble personality, Dr. Dj Mukesh Kumar. He graduated in microbiology at the New College, University of Madras, postgraduated in microbiology at Bharathidasan University, Master of Philosophy in Biotechnology at Bharathidasan University, and doctorate in microbiology, interdisciplinary botany at University of Madras, postgraduate DMLT at Loyola College. He served as principal scientist at Avigan Biotech Private Limited, adjunct professor at Sri Balaji Dental College and Hospital, Bharat Institute of Higher Education and Research, Savita Dental College and Hospital. He worked as post-director, oral researcher funded by UGC at CIS in botany, and assistant professor in postgraduate and research department of microbiology at Azan Memorial College of Arts and Science. He has 6 years of research experience in bioactive microbial metabolites and its molecular characterization, and also worked as lecturer in Department of Microbiology at Tagore College of Arts and Science. Junior Research Fellow funded by UGC, biodegradation of dioxin by microbes. Now he is working as a principal scientist for Spark Biosolutions Private Limited, Chennai. Welcoming you, sir.
Welcome.
Now I welcome our chief guest, Dr. Dj Mukesh Kumar, to take up the session.
Okay, thanks for your introduction. Okay, can I share my screen?
Yes, sir.
Okay. Okay, is it visible? Screen is visible? Can everyone see the screen?
Yes, sir.
Okay.
Yes, sir. It's visible.
Okay. Thank you. Thanks for your nice introduction. So, okay. So it's all about what I did and how I came up. So a lot of research experience. Okay. A lot of good and a certain bad. All those experience made me strong. So regarding this webinar, okay, it is especially about how research can be transformed to patents. So recent years, now mostly everyone focusing on their publications, whatever the research they do. Okay, they go with the publications like publishing in UGC journal, Scopus journal, okay, all standard journals. Okay, but in another hand, okay, there is always a lack about the research what they publish. Okay, how they can transform that research into a valuable patent, like whether it is in process or whether it's in a product. Okay, so there is a lack in that, but they do good research, but still we have lack in transforming that research into patents. So when we ask a question why it is happening, why it is not happening, okay, the transformation, the complete transformation of research into patent means, okay, mostly the lack of a prior knowledge about filing a patent or what to do for patent. So that is the lack making them not to take the publication or the research into a patent valuable product or a process patent. So here I'm just going to going to explain, it's in short, okay, how we can transform our basic research, normal basic research, into a patent, how we can file a patent, okay, how we can take it to a product or a process. That's what we are going to see. And if we are very good in filing patents or publishing patents, okay, what we can do next with that, that's very important. Okay, being a researcher, okay, what and all the opportunities, okay, that we have after taking the research into patent or publication. Okay, that is also we are going to discuss here. So it's like, putting a vinegar churi. Okay, whatever the work we do, okay, like whether it is a mini project or an internship or a major project, okay, so always we'll come up with a research project idea. Okay, normally this kind of ideas, okay, as far as for my understanding, okay, our guide, our mentor, okay, they will give some ideas, some suggestions. For example, if when I started doing my masters, okay, my mentor, Professor P.T. Kalachelvan, he gave an idea, okay, why don't you work in biodegradations? So just he gave a term, biodegradation. So from that term, okay, I called a concept, okay, so what we can do in that biodegradation, okay, what is the gap, what is the scenario, okay, it's happening in the current biodegradation platform. So all those things that, okay, we do some basic table work, basic search, or all those things we'll do. So here also, okay, whatever the field you are, okay, so if you have an idea, project idea, okay, so whether you are a PhD scholar or whether you are MSc, okay, so you start write your project idea, okay, what kind of a thought you have, okay, if you are getting it from your mentor. Recently we were working on nitrate reducing bacteria, okay, that idea was given by the mentor, okay, so what, after getting that idea, okay, what we have to do is, obviously we will go for scientific publishing, and normally, okay, we'll go for scientific publication search. Scientific publication search, it is like what we do, we'll take a topic, we'll go to the Google, we'll search, and we'll start collecting the reviews, okay, or research papers related to our topic, okay, related to our topic. We'll get all the materials, okay, from the Science Direct, from the PubMed, from MDPA, all established journal, we'll start collecting our articles. Okay, then good, that's good. But what I'm trying to say is, okay, while you are collecting, how you are giving importance to collect the data from the journal side, from the scientific publishing side, okay, the same way we have to give our importance to the patent search also. Okay, I don't know how far you are doing patent search, okay, to get the idea of what we have, okay, is it established there? So the same way how you surf Google to download your article. For example, day before yesterday, we were working on dry toilet concept. Okay, so we just typed, okay, what and all the dry toilet concept already existing in the domain. So we downloaded some research articles. The same way we switched over to patent database also. Okay, Google database. There we start typing the same keywords and we started collecting the patent material, which is for the idea what we have, the objective what we have. So what is the use? So as I said, we all know, okay, we can collect materials from Google, Science Direct, PubMed, MDPA. But why we have to search patent? Okay, that is the importance, okay, that I'm going to share here. The idea or the objective, okay, what we have, okay, that will solve the technical problems. that will solve the technical problem. For example, if you are designing, okay, if you want to develop, okay, a water purifier. So the problem is, okay, we have to purify the water. So that is the solution, ultimate solution. Okay. So for that, if we have an idea, okay, before that we have to go for how we do scientific search. The same way we have to do patent search. So the basic importance is the researcher to identify which problem are already solved and which one remain open challenge. So this is the important thing. This is the thing we lack. This is the thing we lack. We always go to patents, we'll go to scientific search. Okay, there we'll try to get a lot of articles, but by come acrossing that articles, okay, we are not sure, okay, what is the exact problem which already solved and what are the remaining open challenges are there that we have to come across. Even by downloading patents from the patent domains, patent database, okay, by reading those contents, okay, we can come across what is their own, what is their idea already exist, and from that idea, okay, we will get a gap, okay, how we can develop our concept, our product, our process on our own way. See, the European Commission estimates up to 30% of their R&D activity is wasted since the technical solution developed already exists. So this is the reason, because if you search, okay, like if you start collecting articles from Science Direct, MDPI, PubMed, okay, these are all easily available domains, okay, that will not exactly give you concept, okay, that where is the gap, where is the scientific gap. But when you go and search patents, okay, see, for example, even this happened to me in the beginning stage. I thought of doing a research on biosolid. So what I did is I started collecting articles, Science Direct articles. So I came up with the idea that, okay, the biosolid concept is very new and nobody is done much research on that. So I started developing, I'm working on it. So when I go for patent filing, the patent attorney said, did you did such patents? Did you did? I said, no. So what she did is, she did patent search and she gave me a lot of articles related to biosolid. And that made me down. Okay, the concept is already exists. So I wasted my time and money. So that's what, okay, when you start developing an idea or an objective, okay, how you give importance to your scientific search, the same way you give importance to patent search also. So what that will give, we have discussed, okay, researching the database to determine if an invention we want to patent has already been patented or not. So that's what we are doing the patent search. Even this will help similar inventions in case there is a need to readjust. That's the point. Okay, if already the concept is filed. Okay, so by come across that filed patents, okay, where we can readjust our concept or our invention, then we can go for a new filing, patent filing. So this will avoid the idea stealing. That means infringement. Okay, already existed and we are copying the same thing. Okay, so for the patent search, initial thing for the patent search, okay, what we need is keywords. How we serve articles from the Google. The same way, Google patent search is also there. Okay, so there we have to, before going to search, okay, we have to coil keywords. We have to write a valid keywords. That means exact keywords with the combinations. Okay, we have to write related to our topic. For example, my field of specialization. One domain is working on collagen wound dressing material. So how I can give topics in Google search collagen wound dressing material. So it will give all the patents related to collagen wound dress. So I have to narrow down herbal based collagen wound dressing material. So all these combinations I have to try, and I will come to know already what and all the existing concepts available there. Actually, when I talk about this, especially when collagen wound dressing material, so I'm very much interested in product development. It is not like working on anti-cancer or anti-inflammatory. So whatever the work I do, I wanted to make it as a technology transformation, because there is more demand and technology transformation. So normally what I do is, when I try to work on some topic, so I'll go to the end product user. For example, in case of collagen wound dressing material, it's a dressing material used for wound burns. Burn wounds we use for better wound healing. So what I do is I'll go and talk to the doctors, because they are the person who use that product on patients. So while I'm discussing with them, they will come across with lot of suggestions, like, Mukesh, the product should be very thin, the sheet must be very elastic, and the sheet must be have some spongy property so that it will absorb the wound exudates. So what I'm telling is, when you're developing a concept, an idea, you just have a thought that who is using that, where it goes. From that, you start doing your research, you start draft your concept, because by seeing by reading few articles, you will have an idea, this is the concept and this is what I'm going to do. No, that is not good. Go to the end user. Then if you come across their suggestions, their value addeds, then that will give a new product which is you can provide, or the concept which you can provide which is not already exist. Or else, if you download materials from the Google Science Direct, it is already there. Then what is the new in your concept? What is the new in your product or process? So that's what I'm telling. Go to the end user, discuss with them, come up with an idea, and with the patent search, you can download full articles, like how we are downloading research papers from Science Direct. So it is not only Google patent, European patents are there, patent search is there. Even we have IPR, Indian Intellectual Property Search Engine is there. You can type, you can go there and you can type your keywords and you can download your materials, what you want to work it out. So once you downloaded the materials, so you have to study the existing concept, existing methodology or existing product, how they develop the product. So after the Google patent search, so you have to write a draft. That means report writing. So for filing a patent or to take your research, for example, if you are a PhD scholar, I'm talking about this concept about the PhD scholars. You have already done research, and you have to take this thing into a product. So what we have to do is the same thing. You have to go to the patent search engine with the help of keywords, download the materials, check your concept is already existed or not. If it is not existed, you have to make a search report, with your keywords, how much percentage of concept it's already available and how much percentage is novel. So with the report, by seeing the report, you will come to a conclusion, your idea, how much percentage your idea. For example, with your keywords, the conclusion says that your idea, your concept is 10%, 20% means the 80% of concept is already existed. Then there is no use of working on it. Okay. So the more percentage of uniqueness or novelty, okay, that helps you to bring up a new product process patent. So as I said, first search report. So once you get the search report, okay, then drafting. So drafting, it is like writing a research paper only. Okay. So for that, okay, we have a lot of service providers. Okay, even Avijan, we are providing a very good service provider for patent search, drafting, and patent filing. Okay, I'll come and discuss this. Okay, at the end of the presentation. Okay, so report writing. Report writing means it's like writing a research paper. Okay, see, after the patent search and report writing, okay, there are two options. One is if you are a beginner in research, okay, and you want to discuss this concept with many people. Okay, so what I suggest is you have to go for provisional filing. You have to go for provisional filing. Provisional filing means a temporary, okay, a temporary filing, okay, which will give you 1 year time. Okay, the provisional validity of the provisional filing is 1 year. So you can go for provisional filing with the 1 year. Okay, if you are very sure that you can complete your research, okay, at the end of your provisional filing, you contact your patent attorney. Okay, they will help you to file a complete patent. Okay, so this is for the beginner. Okay, but some people, some researcher, okay, I have done everything. Okay, I have a concept, I have a result, okay, everything. So what you can do is you can go for patent search. So after the patent search, okay, you will get a report. Okay, so from the report, if the novelty is more high, okay, you can go for drafting, patent drafting, and after that you can go for patent filing. So this is how, okay, I suggest how importance you give for collecting the articles from the Science Direct. The same way, okay, if you want to filing, okay, if you want to take it your research to patent, okay, you have to give importance to the patent search also. So this is the basic idea I have given, okay, how we can transform our research into a patent. Okay, the main thing is first, whatever the concept, whether you have a scrap or whether you have done everything, okay, you go with search report. Okay, first you have to go for patent search by using the database, patent database, use your keywords, collect n number of articles related to your concept and check the novelty. So when the novelty is good, it is easy for filing and it is easy for granting also. If the novelty is less, we have to struggle a lot for filing, and granting becomes more hard. Okay, so as we all know, why we are doing this patent filing, okay, and a very important thing is recent days many people are asking this question, sir, if I file a patent, okay, can I publish that work as a paper? Yes. Okay, but the thing is, once you publish your research paper, you cannot file that work as a patent. Keep it in mind. Once you publish first, you cannot file the concept as a patent. But if you file the patent first, okay, you can redraft it slightly and you can publish that work also. It is like one work, two benefits. Okay, but the purpose of filing a patent is keeping your concept secret. Okay, but when it comes to academics, okay, when I talk about academic research, okay, so all are asking that, okay, sir, I have to take it my patent as well as to the publication. So you can do both also, but once you publish, you cannot file the patent. Okay, so why we are patenting is, okay, this is for startups. Okay, so nowadays, okay, we are coming up with a lot of incubation centers. Okay, everywhere we have incubation centers. Even NAG is recommending, okay, why don't you have incubation center in your college, in your university? What is an incubation center? Okay, the center provides you all the facilities, okay, if you have a great idea to make that idea into a product or a process. So if you have filed a patent, okay, if you have processed a patent, you have a lot of opportunities, okay, and a promising opportunity, okay, to make your concept as a startup. So for that startup, okay, everybody will not have money, okay. So for the startup, okay, incubation center helps you to make your dream come possible. Okay, the idea what you have, the concept what you have, okay, which will provide you all the support, basic supports, okay, to make your concept into a viable commercialized product to make a startup. That's called TBI, Technology Business Incubator. Most of the universities nowadays, they are having TBI. So what TBI will provide? TBI will provide you a basic investment support, infrastructure, office, technical service, and they will provide you training in both the end. Okay, and they will provide you basic funds. And once everything is good, okay, they will help you to bring investor also. So if you are an inventor, okay, TBI will help you to bring the investor. So no, there are a lot of grants facilities are also there. Okay, if you are going for patenting, okay, because everywhere they are asking, okay, if you have a patent, okay, if you have a grant patent, okay, they are ready to help you for your startup, okay, and they will pushing you to become an entrepreneur. So India have a lot of facility. No, in India, okay, even central government are providing a lot of supports for helping you for the startups. And this shows okay, how for state-wise, okay, the TBIs are helping to come up with an idea. And when it comes to Tamil Nadu, okay, so Tamil Nadu startup policy, if you go and search, okay, there they are providing a very good support for startups, okay, especially from Tamil Nadu. And Venkat Raghavan, okay, he's the person who is helping to make that reality come true. And government agencies supporting for TBI. And what are all the fields, okay, they are helping you for the startups. And so how they are helping you? No, so as I said that if you have a concept, okay, so when I talk about my research, okay, so it is a product based. So when it comes to product based, there are two categories. One is a new one, the product which never exists, okay, that's a new one. And the second one is the product is there, okay, but based on the demands, based on the requirement, based on the suggestions, okay, we are improving that. That means value added products, okay. When the product is novel, new, okay, that is considered as a new novel product, okay. The product is already there, okay, but based on the demands, based on the requirements, okay, we are improvising means, okay, that's become a value added product. So that's the difference between innovation and product development. And I know I am also associated with EcoPro Auroville, okay, there I am a consultant. We are developing a lot of concept to make the rural development. And this was one of our concept, dry toilet concept. Okay, this is a patentable. And in and around Auroville near Dindivanam, we have constructed a lot of dry toilets to make people to better use, and by converting our fecal material into composting manure. And when we talk about collagen dress wound healing material, okay, this is another domain. Okay, so recently we have published our a patentable product. We have published in Indian patent website. So when we talk about collagen wound dressing material, there are a lot of types. Okay, sheet, powder, gel, and sponge forms are those. These are all the way we have developed the products. These are all piece of work of our experiments. So this one we got a fund, and with the help of that, okay, we have filed our first patents. And this is our 2020, we have filed 3 patents. Recently it was published. So when it comes to product, okay, what should, okay, when it comes to invention, how should. So you have to understand this concept. So industry, Petroplast, Accord, Orchid Pharma, they are the people, those who are supporting us for making our proposals into taking our proposal to the next level. So with this concept, we may think that, okay, what we plan, it goes very smooth, but the reality, it is not like that. Okay, I just simply said that, okay, how we can transfer our idea into a patent. Okay, so understand the basics. Okay, so when it comes to reality, it is a bit hard. Okay, and we are there to support you. Okay, your idea, a small idea, can change the world. So Avision Biotech, we are here. Okay, we are having a very good infrastructure facilities. And when it comes to research into patent, because see, that's what, okay, we do research and we proved ourselves that we do patents also. So that's why I said, okay, we do research and we do patents. So that we are providing, okay, we are helping you to finding the search report, and we are helping for drafting your concept, and we are helping for taking you for patent filing also. And we are we are ISO certified, and we have MSME certifications. Okay, so that it proving that, okay, we are not just like a company. Okay, we are a promising company that will ready to help you to take your research into a patent. Thank you. Any questions?
Any questions?
I expect a lot of questions. நீங்க தமிழ்ல கூட கேட்கலாம், no problem. லாங்குவேஜ்ன்றது புரிஞ்சிக்கணும், அவ்வளவுதானே. உங்களுக்கு டவுட் இருந்தா தாராளமா கேட்கலாம், எந்த டவுட் வேணாலும் கேட்கலாம்.
Rakesh?
Feel free and come and visit our laboratory. There we do extensive research. And if you have any idea to develop a product or a process, okay, please contact us. We have shared our contact details. Okay, so you can contact, so we can help you to frame your concept into a product. When it is possible for us, even it is possible for you also.
In chat box, there is one question they asked.
Yes.
If already the microorganism is already existing one, in that from
Yes.
that we are getting any product, means we can get patent for that organism or for the product alone, sir?
Actually, if it is a microbe, okay, we cannot go for we cannot file the patents for the microbes if it is unless until GMO, genetically modified. If we are recombinant, with the help of recombinant, if we modify the organism, we can go for patents. But in that case, we can, yes.
So the product which is isolated from the microbes, we can get the patent.
Yes, yes. We can do both the filing. No, the process filing also we do, and the product filing also we can do. That's possible. Even the most of the drugs we are filing in that way only. Derived from the microorganisms.
Okay, sir. Any other questions? Sir, another one question.
Yes.
Before we are starting our project or anything, just idea alone, we
Yes.
can get the patent, sir?
Yes. Very good question. Really very good question. Okay. Idea alone we can. Okay, because see, we are in a year, era, we are thinking very slow. Okay. after 20 years, after 50 years, okay, the idea, the concept, even people are ready to buy our idea. People are ready to pay money for our concept. Okay, there are a lot of investors are there for taking our idea into a product or a patent or process. It's possible. It's possible. 100% possible. For that only, we are encouraging startups. Do you understand now? So if you have an idea, okay, so what TBI is telling is, come here. We are help you. We are here to help you. Come with the idea. Even IIT, they are providing a lot of basic funds for college students. Okay, even Avision, we are helping a lot of people to transform their idea into a startup. Even we have a TBI facility. We have a lot of we have a MOU with IIT. Okay, there we are taking the concept into MOU, so that they are getting basic funds. It is not like targeting for only TNST. Okay, your idea can be transformed into startup. Come up with the idea. We are here to help you.
Yeah, sure. Thank you. Any other questions?
Okay, ma'am. That.
Aishwarya, continue.
Yes, ma'am. Thank you, sir. We are
Thank you,
very
ma'am.
with your words and gain more knowledge from this topic. Now I would like to call upon D. Muttarasi to deliver the vote of thanks.
Good evening to all who all are gathered here on this wonderful evening. It gives me an immense pleasure to deliver the vote of thanks for this event to all dignitaries assembled here. First and foremost, I thank our respected chief guest, Dr. Dj Mukesh Kumar, Principal Scientist, Avanjan Biotech, who despite his busy schedule has found time to grace this occasion. We are grateful to thank our Honorable Chairman, Dr. K. Vasudevan, for his words of encouragement. His able guidance as always encourage us. I thank our well-wisher, Vice Chairman, Engineer V. Prasanna Venkatesh for this support, vision and commitment. And also my sincere thank to our motivating principal, Dr. Uma Bhaskar, to support for us. I thank our SHEE Club and IIC coordinator for arranging such a motivational and informative session. I'm so grateful. Thank to our staff members. Last but not least, thank to our students for making the session a good success. Thank you.
Thank you, ma'am. Thank you.
Thank you.
Thank you, Muttarasi. Now I would like to call upon M. Kirtika Devi to end up the session with National Anthem.
जनगण मन गति नायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्छल जलति तरंगा तब शुभ नामे जाहे तब शुभाशिष माहे गाहे तब जय गाता जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
Thank you.
Thank you. Thank you. I end up the session.
Okay, ma'am. Thank you, ma'am.
Thank you.